Interphase is the period of cell growth when the cell makes new molecules and organelles. In late interphase, or G2, the cytoplasm contains two centrosomes, shown in orange. Within the nucleus, the chromosomes are duplicated, but they cannot be distinguished individually because they are still in the form of loosely packed chromatin fibers. Each chromosome then consists of two identical sister chromatids.
As the chromosomes coil up, special proteins cause the homologous chromosomes to stick together in pairs. The resulting structure has 4 chromatids. Within each set, chromatids of the homologous chromosomes exchange corresponding segments. This is called crossing over. Crossing over rearranges genetic information. As prophase 1 continues, the chromosomes coil up further, a spindle forms, and the homologous pairs are moved towards the center of the cell.
At metaphase 1, the homologous pairs are aligned in the middle of the cell. The sister chromatids of each chromosome are still attached at their centromeres, where they are anchored to spindle microtubules.
Notice that for each chromosome pair, the spindle microtubules attached to one homologous chromosome come from one pole of the cell, and the microtubules attached to the other chromosome come from the opposite pole. With this arrangement, the homologous chromosomes are poised to move towards opposite poles of the cell.
The attachment between the homologous chromosomes of each pair breaks, and the chromosomes now migrate towards the pole of the cell. In contrast to mitosis, the sister chromatids migrate as a pair instead of splitting up. They are not separated from each other, but from their homologous partners.
In telophase 1, the chromosomes arrive at the poles of the cell. When they finish their journey, each pole has a haploid chromosome set, although each chromosome is still in duplicate form. Cytokinesis occurs along with telophase 1, and 2 haploid daughter cells are formed.
Meiosis II is essentially the same as mitosis. The important difference is that meiosis II starts with a haploid cell that has not undergone chromosome duplication during the preceding interphase. During prophase 2, a spindle forms and moves the chromosomes towards the middle of the cell. During metaphase 2, the chromosomes are aligned as they are in mitosis, with the microtubules attached to the sister chromatids of each chromosome coming from the opposite poles.
In anaphase 2, the centromeres of sister chromatids separate, and the sister chromatids of each pair move towards opposite poles of the cell. In telophase 2, nuclei form at the cell poles, and cytokinesis occurs at the same time. There are now 4 haploid daughter cells, each with a single chromosome.